Hello and welcome to this video on how DNA is extracted. Unlike what is shown on TV shows like CSI and Law and Order, DNA is not readily extracted and then typed from small amounts of blood or degraded samples. In fact, extracting a good DNA sample is much harder. Most human tissue has some sort of DNA. The exceptions to this are either dead cells or products like hair follicles that are pure protein. Other examples are cells that are going to make a one-way trip, such as red blood cells. This means most cells have some kind of DNA. The same is not true of all bodily secretions. In fact, this applies to all living organisms, where plant cells, bacteria, and viruses have DNA, but not the fluids within. As has been described previously, the polarity or isoelectric point of many things is variable. In the case of DNA, the overall positive or negative charge of the sample depends on the constitution of the sample. Does it have a bias towards guanine, cytosine, adenine, or thymine? One or more of these can shift it to being slightly positive or slightly negative. Generally speaking, DNA is negatively charged. The benefit of this polarity is that it makes extracting crude DNA samples very easy. One of the most common extraction methods uses this polarity and density of DNA. It follows this procedure that you can do at home. Take a relatively large amount of whatever you want to make a sample of. This could be leafy greens, lentils, even meat. Blend it, including a small amount of salt, about 1 milliliter, along with 200 milliliters of water. Do so for about a minute, preferably a little bit less. This will create a very thin mixture of whatever you've attempted to create a DNA extraction of. You then add a small amount of detergent, about 30 milliliters. Mix it and then let the mixture sit for 5 to 10 minutes. Divide this into test tubes, each one being about a third full. You then want to add an enzyme to this test tube and mix it slightly. This can be anything that you buy in the supermarket, such as a specialized enzyme, or if you are really pressing to find an enzyme, try adding something like pineapple juice. Take your test tube, tilt it to the side gently, and add 70 to 95% alcohol, either isopropyl or ethyl alcohol. Add it slowly and gently until it forms a layer on top of the test tube at about a 1-to-1 ratio with your original sample, meaning the test tube should be about 2/3 full. After this sits for a little while, the alcohol should force the DNA to come to the point where the alcohol and water meet. The alcohol is slightly different in density to the water, which means it sits on top. However, the DNA is awkward and will only sit in between the two phases. This will create clumps of white stringy stuff. You can then siphon off the alcohol component, and this will expose the DNA and allow you to separate it out. In a laboratory, it is slightly different but similar, as it relies on specialized reagents. The method we are most familiar with is called the phenol chloroform extraction. It extracts RNA as it is the mediator of active genes. DNA is converted into RNA when the cell wants to use it. This then tells you what is happening at a genetic level. This can then be tested using something like a cDNA synthesis kit to create a picture of how a cell is working and what genes are being expressed, which is done with a PCR reaction that will be described in another video. To extract RNA, the phenol method is used approximately as follows. Your samples of tissues, cells, plant matter, or whatever else you are looking into will have been blended or otherwise broken up to expose the insides of the sample's cell, particularly the nuclear envelope. You add this to your combination of phenol, chloroform, and isoamyl alcohol, generally at a ratio of 25 to 24 to 1. In other words, 50% phenol, 48% chloroform, and 2% isoamyl alcohol. The combination of these chemicals and your sample is shaken for about 20 seconds. It is then centrifuged for 5 minutes. This leaves a thin aqueous layer at the top, which needs to be removed with pipette and put into a new tube. You do not want to carry over any of the pink fluid that is phenol during this process. You now have what should be a relatively pure sample of RNA from your sample. It needs to then be isolated using the ethanol precipitation method, although there are some others available. The tubes are kept in a freezer overnight to aid in the extraction. You then centrifuge them the next day for 30 minutes to produce a small, barely visible pellet of RNA at the bottom of the tube. This pellet is what you are ultimately after, but as it is now cannot be used. You need to remove all the other fluid in the test tube except for that pellet. You then add a small amount of ethanol, which is used to wash the sample. This is then spun in the centrifuge again for another 2 minutes. After this, you remove the ethanol and repeat the process a number of times again. This takes away any residual phenol or other contaminants from your original extraction. You then allow the test tube to either remain open on the bench for a time or in a specialized drying device that will dry out your RNA pellet. This is then resuspended in microliters of a buffer solution or a carrier of your choice. Now, just adding the carrier will not help you with this. You need to break up the RNA pellet by pipetting the entire solution up and down repeatedly. The entire process relies heavily on differences in density and polarity, which is why you are able to isolate and extract the RNA and then purify it. This then leads to a mixture of your RNA or DNA and ethanol or similar contaminants. Far fewer contaminants than the homemade version, but there is still going to be something. In an ideal world, you can centrifuge and wash the RNA extracted until there is almost no contaminants. The way to know how this is achieved is through a device like a nanodrop or similar spectrometer. It shines a specific wavelength of light that DNA and other nucleotides correspond to. It will also pick up other content such as ethanol. If it reads a low nucleotide sample but high in other wavelengths, you know it is a dirty sample that will produce poor results later. Understand, this entire process does produce tiny amounts of RNA, and as a consequence, any cDNA is going to be in tiny quantities, something in the range of nano to picograms. Hopefully by doing your own DNA extraction, you can visualize it for yourself. Each PCR reaction, if you were to go down that route, requires at least 20 nanograms to be a reliable result. Getting this much from a random sample found in a crime scene, such as that in TV shows, is almost impossible. Even if you could get that much, the chances of it being degraded beyond use are extremely high. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.